Welcome back, friends. Good evening. So in this session we are going to see the previous year questions from the chapter molecular basis of inheritance. So let's go into the session. You can see the lac operon consists of. So this question was asked in 2010. You can see the options: 4 regulatory genes, like 1 regulatory gene, 3 structural genes, likewise 4 options has been given. If you know the structure of the lac operon, this answer will be very easy. So actually this is the direct question from the book. If you see the structure, you can clearly identify. See, this is the structure of the lac operon. So you can see the answer. You can see what is there: 1 regulatory gene and 3 structural genes. So which is the regulatory gene here? You can see. So this is the regulatory gene. So 1 regulatory gene was there, and here you can, this side you can see from here to here, lac Z, lac Y, lac A. Totally 3 structural genes are there. So totally 1 regulatory gene, 3 structural genes. So the option B is the correct answer. So you can see here, 1 regulatory gene, 3 structural genes. Option B is the correct answer. Let's move into the next question. Here you can see the 3' to 5' phosphodiester linkages inside a polynucleotide chain. So to join, what will be a polynucleotide chain? So many nucleotides will be joined together to form a polynucleotide chain. So 2 polynucleotide chains together give the structure of the double helical DNA. Now the question is, in single polynucleotide chain, the phosphodiester bond linkages will be present where? They will be joining which component? Whether it will join one DNA to another DNA strand, or nucleoside nucleoside, nucleotide nucleotide, or nitrogen base with pentose sugar. So this also, if you just see the structure, easily you can answer. You can see the structure here. So this is the structure. So here easily you can see that. So nucleotides will be joined together. Yeah, you can see this is nucleotide 1 and this is nucleotide 2. So 2 nucleotides will be linked. You can see in the next, with the ligase, with the help of the ligase enzyme, you can see the phosphodiester bond. See the pink color, this is the phosphodiester bond. So the bond is found between the nucleotide 1 and nucleotide 2. So this is the second nucleotide. So it will be going like this. So the polynucleotide comprises many nucleotides. See, all the nucleotides will be linked by the phosphodiester linkage. So the answer will be, you can see, 1 nucleotide with another nucleotide. Option C. You can see the option C is the correct answer. So you can see the here more clearly. See the yellow color. The yellow color shows the phosphodiester bonds, phosphodiester linkages between one nucleotide and another nucleotide. So this will be between the first and second nucleotide, and here between the third and here it will be having, see here, so this is the first phosphodiester bond, this is the second phosphodiester bond. So why it is diester? Here one bond and here one bond, so totally 2. So diester. Again here one and here one, so totally diester, phosphodiester bonds. So 1 nucleotide with another nucleotide linked by the phosphodiester linkage. Let's move to the next question. Cite the 2 statements out of the 4 given below about the lac operon. So you have to identify the answer from this. So glucose or galactose may bind with the repressor and inactivate it. So as you all know, the glucose acts as what? The glucose or galactose acts as the energy molecule. When the lactose is broken down into glucose and galactose with the help of galactosidase enzyme, so that will be utilized as the energy molecule by the bacterial cell. So the repressor will not be inactivated, okay, with the glucose. That is wrong. The second thing, in the absence of the lactose, repressor binds with the operator. So lactose means that is the inducer here. In the presence of the lactose, the lactoperon will be switched on. In the absence of the lactose, what will happen? The repressor surely will bind to the operator region. So second option is correct. And the third option, you can see that the Z gene codes for permease. So Z gene will not code for permease. The Z will be coding for what? Galactosidase enzyme. So it is also wrong. The fourth option, so this was elucidated by Jacob and Monod. So that is right. So option 2 and 4. So you can see the answer will be C. So the C will be the answer. These 2 and 4. You can see the structure more clearly, you can understand here. You can see the structure. By seeing the structure, you can easily understand which will be the right answer. So the LAC-Z codes for what? You can see LAC-Z codes for beta galactosidase. So beta galactosidase, you can see. But they given permease. So that is wrong answer. Okay. So the option is C. 2 and 4 will be correct. So in the absence of the lactose, the repressor binds with the operator region. You can see in the first A diagram, in the absence of the lactose, what happens? The repressor binds to the operator region. So that is right. And the next is elucidated by Jacob and Monod. So option C is right. Next question. Which one of the following does not follow the central dogma of molecular biology? What is my central dogma? It is the main process, main principle in which all the living organisms are living. They are surviving with the help of this central principle, that is from DNA to DNA, that is replication. From DNA to RNA is transcription, and then RNA to protein is called as translation. This is the central dogma. You can see here DNA. From DNA, RNA will be produced. That is process called as transcription, and from RNA, protein will be formed. That is called as translation. This is central dogma. The question is, which among the four does not follow the central dogma? You can see here the P, first option, that is a plant. So plant follows central dogma. All the plant species follow central dogma. And the second thing is mucor. What is mucor? That is a fungus. So all the fungus follows the central dogma. And the option C is what? Chlamydomonas. That is algae. So all the algae follows central dogma. The last option was what? HIV. So HIV is what? It is a virus. It is called as retrovirus. So in this case, the genetic material of the HIV virus is RNA. Here it will not follow the central dogma. The reason is, you can see the picture here, the HIV is a retrovirus. It is not follow the, you can see the, they have the genetic material as RNA. So from RNA, what will happen after entering into the host cell like humans? They will produce DNA by the process called as reverse transcription with the help of the enzyme called as reverse transcriptase. Then again the DNA will be producing RNA, that is transcription, RNA to produce translation. So in HIV, the normal central dogma will not be followed. It will be in different manner. It starts with RNA and ends with protein. So the option D will not follow the central dogma. The next question. What are the structures appear as beads on the string in the chromosome viewed under electron microscope? So as you all know how the DNA is packaged inside the nucleus of the cell compactly with the help of the protein, structural proteins called as what? Histone proteins. So when you are viewing under the electron microscope, it will be like beads on the string. So what are those beads on the string structure they are asking here? Whether it is a nucleotide or nucleoside or base pair or genes. So you can see the structure, you can easily identify here. You can see. So this is the beads on the ring structure. So many beads are visible in the electron microscope, so that you can see this black arrow. This black arrow is showing the bead, like round structure, dot is there, no black dot. So that is called as nucleosome. Whereas this white arrow, that is showing the linking between the two nucleosomes, that is the linker DNA. So the white arrow shows linker DNA, black arrow shows nucleosome structure. So nucleosome will be there, you can see here. So between the one nucleotide, one nucleosome, another nucleosome will be linked by the linker region. So this linker was shown as white and this was shown in the black. So this is the beads on the ring structure. So based on this diagram you can easily answer. What is that? That is nucleosomes. So nucleotides will be present where? In the polynucleotide chain. That is ATGC will be the right, adenine, guanine, thymine, cytosine. And base pairs will be the between the double helical DNA structure. A pairs with T, G pairs with C. Genes are the functional units, expressible units which is present in the DNA. So those three will not come. So it is nucleosomes is the correct answer. Next question. Unequivocal proof of the DNA as genetic material came from the studies. Actually this is the direct question from the book. Unequivocal. What do you mean by unequivocal? Means what? Unequivocal means like without any doubt. Okay, 100%. The proof is DNA is a genetic material, no other thing. Only the DNA is a genetic material, is not a protein, is not RNA. So that proof was given by, based on which study they are asking. Okay, whether it is bacterium, it is fungus or viroid, it is bacterial virus they are asking. So it is a direct question from book, NCERT book. You can see I have given the screenshot of the 6.2.1, that is a genetic materials DNA. In 6th chapter you have given, right? So here you can see in which organism they have performed the experiment. You can see here it is bacteriophage. So bacteriophages, that is, what do you mean bacteriophages? That is a virus which infects bacteria. So that is bacterial virus. So the answer will be what? Bacterial virus. So based on this experiment only, the proof given like DNA is the genetic material without any doubt. And next, if one strand of DNA has nitrogen base sequence ATCGTG, then what would be the complementary RNA strand sequence? So it will be very easy actually. So you can see here I have given the pairing in the strands follow the pairing principle. The A will pair with what? Usually in DNA means A will pair with T, G with C. Here they have asked what? You can see they have asked about the complementary RNA strand. In RNA you won't have thymine. Instead of thymine you will having what? Uracil. So based on that you have to answer. So the question they have given is ATCTG. So in the first A will be pairing with what? T actually. But in case of RNA, instead of T, what you will get? U. So A, U. And the second thing, T will be pairing with A. C will be pairing with G. Again T will be pairing with A. G will be pairing with C. So only in case of RNA, you will get what? This U will be there. Whereas in DNA, this is DNA. And DNA you will not get. So in DNA you will not get what? That uracil. Only in RNA you will be getting this uracil. So the RNA will be what? This is the answer. UAGAC. So answer will be UAGAC. Next question. Transformation was discovered by. So this is also the direct question from the book. So what did Michelson and Starr did? What Hershey and Chase did? Griffith, Watson and Crick. If you know all these scientists what they did, easily you can answer. So Michelson and Starr, what they did? They did this semi-conservative model of replication was given by Michelson and Starr. Whereas Hershey and Chase, what they did with the help of bacteriophage, they given the proof that DNA is a genetic material. And Watson and Crick, they give the double helical structure of the DNA. So Griffith only, with the help of the Streptococcus pneumoniae, they have given the transformation principle. So answer will be Frederick Griffith. So I can given the screenshot. You can see here the transforming principle, that is called as transformation. The process is called as they have performed. So Frederick Griffith. So answer will be Griffith. Next, which of the following is required as an inducer for the expression of the lac-operon? Previously in some question I have said you that lac-operon. In the name itself we have what? Lac. So lactose acts as the inducer. So you can see the diagram here will be, this is also directly from book only. You can see here, directly from book. Easily you can answer this actually. See at the last they have given, right? So inducer, you can see here. So lactose is a substrate for the enzyme beta galactosidase. It regulates the switching on, switching of the operon. When lactose is present, it will be switched on. When lactose is absent, the operon will be switched off. So it only induces the operon to be on. So it acts as an inducer. So lactose is the answer. So experimental proof for semi-conservative replication of the DNA. So this also I have discussed 2 questions before, I think so. Yeah, semi-conservative model was given by Messelson and Starr, but in which organism first? That is the question here. Whether it is fungus, plant, bacterium or virus. So you can see here experimental proof, you can see it is all direct question from the book. So it was given in, you can see, E. coli. E. coli is the bacterium. So bacterium only first, you can see that was the first experimental proof for the semi-conservative replication. So bacterium is the answer. The next question, select the correct match. You can see here the Alec Jeffrey, Messelson and Starr, S.A. Chase, Jacob and Monod. So easily you can find the answer. The Jacob and Monod, they only give the lac-operon model. So option D is the right answer. So what did the Jeffrey, Messelson, Starr, HHS did? You can see here. See the Jeffrey, what he did now. Alec Jeffrey, his full name is Alec John Jeffries. So he only given the like genetic fingerprinting and DNA profiling was discovered by him only. And then Messelson and Starr experiment, what they did, semi-conservative mode of replication was given by them. Hersey and Chase proved the DNA as a genetic material with the help of bacteriophage virus. So these 3 are given here wrong match. So first 3 will not come. So last option is the right answer. So what is Streptococcus pneumoniae? So this was used by Frederick Griffith in his experiment of transforming principle, that is transformation experiment. He used the organism. The Pisum sativum is used by the Grieger, Jogan, Mendel in the genetics, like dihybrid cross, monohybrid cross. In this experiment he used this Pisum sativum. The TME, tobacco mosaic virus, it causes mosaic disease in the tobacco plants. So the last option is the right answer. See here, you can clearly see. Yeah, this is also directly from book only they have given. Jacob and Monod proposed the operon model. So that is the right answer. And next question you can see here. All of the following are the part of operon except. So in operon structure, if you see, you can easily identify. Previous slides I have shown you the structure of the lac-operon in the previous slide. Wait, I will show now. You can see here, you have what? Operator is there, promoter is there, repressor is there. So here repressor, operator, promoter, all those things are there. Whereas you cannot find enhancer in the structure of lac-operon. You can see here, you cannot find any enhancer in the structure of the lac-operon. Yeah. So the answer will be what? Enhancer. You can see here. Yeah. Operator region is there because there only the switch off, switch on mechanism. The binding of the repressor protein will be happening there. So operator is present in the lac-operon. And then the structural genes, they are the main genes. In the first question I have told, right? 3 structural genes will be there, Z, Y and A. And promoter region is there only the enzyme RNA polymerase binds and transcribes the genetic material gene into RNA. So those three will be there, whereas enhancer will not be there. What do you mean enhancer? You can see here. Enhancer sequences or the regulatory DNA sequences. These sequences are bound by specific proteins called as transcription factors. The binding of these proteins will enhance the transcription of the associated gene. That means wherever these sequences are present, they will enhance the transcription. If the transcription is occurring very slowly, if these enhancer sequences are there, what will happen? The transcription process will be enhanced. The associated gene will be there, right? So the gene transcription will be enhanced by these sequences. So in operon structure we cannot find this structure. So the option is B, enhancer. So enhancer we cannot find in the operon concept in the structure. The next, they have given the sequence of the coding strand of the gene. What will be the corresponding sequence of the transcribed mRNA? So what is the coding strand? So you can see here the coding strand is the one that codes for the mRNA. What will happen means the same nucleotide sequence will be there in the mRNA except thymine will be replaced by uracil. So this is the main thing you have to keep in mind. Coding strand. So template strand will be there, coding strand will be there. This coding strand, what it will do means the sequence will be exactly same, the nucleotide sequences. Instead of T, you have to replace by U. You can see here AGGTAT CGCAT. So it is the first sequence that was present, that is the coding strand. You can see the first sequence. So that is the first sequence that is given in the question. So what is coding strand? I said, right? Now what happens? You have to replace T with U. You can see here U and again T with U and last again T with U. So we have to replace all those things. So when you replace the 3 places you have T, 1, 2 and 3 places you have T. So these T's has to be replaced with U. So then you will get the answer. So answer will be what? AGGUAU CGCAU. So this is the answer. So whenever in the question if they give the coding strand sequence and if they ask the mRNA sequence, easily you can write instead of T you have to replace U and directly you can write the same nucleotide sequence and you have to replace with U instead of T. So you will get the answer. Keep in mind, definitely these type of questions will be repeating. Let's move to the next question. The final proof for the DNA genetic material came from the experiments of, final proof, that is what I have said in the previous question. Previous some year also they have asked the question, unequivocal proof. What do you mean that? Without any doubt. Who did the experiment? As you all know, Hersey and Chase. So they only did the experiment. So what about this Avery, MacLeod and McCarthy? Who are they? So they only gave the biochemical characterization of the transforming principle. So what about Hargobind Khorana? So he was involving in the researches like protein synthesis, the importance of the nucleotides in the protein synthesis and genetic code. So his experiments are based on the genetic codes and nucleotides like expression, protein expression. Whereas Griffith, what did Griffith did? For the transformation principle was did by the Griffith. So biochemical characterization was done by Avery, MacLeod and McCarthy. So Hersey and Chase is the right answer. They only gave the proof. You can see here, this is directly from the book only. So the option is Hersey and Chase. So based on this bacteriophage experiment only, they found that DNA is the genetic material without any doubt. Next question. The association of the histone with the nucleosome indicates what? DNA replication is occurring, DNA condensed in chromatin fiber, DNA double helix is exposed, transcription is occurring. So what is the use of histone? I have said in my session. So histone proteins are structural proteins that is used for compactly packing the DNA in the nucleus. So easily you can say the answer, the condensed form of DNA into a chromatin fiber. So that is the answer here. So you can see the histone proteins. You can see here the histone proteins. See that this is the histone 1. So it links the nucleosomes. Nucleosome 1, 2 and 3 are there. So this is nucleosome 1 and this is nucleosome 2 and this is nucleosome 3. So 3 nucleosomes are there. So in between it is linked. You see this is the linking is there, right? So this linking is H1 histone protein. Again here also you can see this is H1 histone protein. So totally what happens, this 8 histone octamer will be there. They will be from the nucleosome structure, and then this H1 will be linking between one nucleosome and another nucleosome. So what happens, they will be compactly packed to form the chromatin structure. You can see here the chromatin structure. You can see here chromatin. So the answer will be what? DNA is condensed into a chromatin fiber. B, that is the correct answer. See here, it condensed into a chromatin fiber. Thank you friends. In next session I will be discussing about more questions from NEET point of view. Thank you friends. Thank you for watching this video.